It's just a science project.
Silent breed is people!
You know, a doctor friend once said the same thing to me. Frankenstein was his name.
It's alive! It's alive! It's alive!
That sounds like something out of science fiction.
Please explain to me the scientific nature of the whammy.
We live in a spaceship, dear.
So? Yes, science!
Program complete. Enter when ready.
Hello and welcome to episode 219 of Science on Top. Today is Sunday, the 20th of March, 2016. I'm Ed Brown and I'm joined tonight by Penny Dumsday.
Hello.
Lucas Randall.
Hello.
And Dr. Shane Joseph.
G'day.
So today we'll be talking about Tyrannosaurus Rex and how it evolved from the size of a horse to one of the largest land carnivores of all time, bacteria that eat plastic, and how what your parents ate before you were born could affect your risk of obesity and diabetes. Thanks a lot, Mum. But let's talk instead about polyethylene terephthalate, better known as PET. It's the most common kind of plastic, and pretty much all plastic packaging and bottles are made from it. And of course, most of that ends up in landfills and waterways. But now a team of Japanese researchers have discovered a new species of bacteria that produces a never-before-seen plastic-eating enzyme. This could be a really big deal, Shane.
Yeah, this is actually really cool. To be clear, there have been other organisms that have been found to degrade plastics, but they've usually, in the past, they've been fungi, and they're apparently very, very hard to grow. Whereas in this case, these Japanese researchers, from what I've gathered from the story, basically just sort of hunted through landfill, took some samples and just essentially chucked them into microbial media and grew these things and found that they could actually degrade plastics within about sort of 2 weeks. And they went a step further, that they actually went and looked at the enzymes that are degrading the plastic. Now, the reason, one of the reasons plastics are so durable is because there aren't really any biological systems to degrade them, because they're produced artificially in a sense. Like, to produce plastics, we use it's like a heat treatment in the presence of a catalyst, which makes long chains of molecules that are basically indestructible. Which is why they hang around for hundreds of years, or they can. And because they've only been developed in the last since about 70 years ago, nothing that we knew of could break it down naturally until now. And they looked at the enzyme that can do this. And what happens is there's an enzyme called PETase, which is basically what breaks down the plastic polymer, like the chain. And it breaks down to a smaller component called MHET, which is then used to produce something else that the bacteria can use as an energy source. So it's a two-step process. What's interesting is that the second enzyme is quite common, or it's been found in other things. But as far as they're aware, they haven't seen this first enzyme, the PETase, before in anything.
Okay. And so you said it takes 2 weeks to break down.
Yeah, I think that it was like a bottle, like they took a plastic bottle. A few weeks later, the plastic was all gone. So it's actually still a pretty slow process. It's not.
So after that 2 weeks, you're left with nothing. No,
essentially, nothing.
Waste product or anything.
Yeah, I mean, probably the bacteria will break this down into ethylene glycol and an acid that they use as an energy source, and then that will obviously be converted into something like carbon dioxide or whatever. That would just be emitted, I guess. So there is obviously, it doesn't just disappear. It is converted into something that's a gas or something like that. But essentially it uses it as a food source. Now, why hasn't this been found before? Probably because no one's gone looking for things that can do this. What's interesting is that that's what they grew. What I want to know is what else can't they grow that does this? There's probably heaps of stuff out there that, and one of the ways they've sort of thought this might have happened is sort of like horizontal gene transfer, that like this bacterium might have developed this one enzyme in the last 70 years or so, which is possible. It can happen. It might just be like an editing of a mutation of another enzyme.
Yep.
And then it's acquired this other enzyme that produces it, that converts it into this MHET molecule, which, that then gets converted into the ethylene glycol. They've acquired that gene through horizontal gene transfer. So now they've got this plastic degrading suite of genes.
But the fact that there is that suite of genes is probably the more exciting thing, is because now we can take those genes, presumably we can put them into say, some E. coli or some fairly simple, straightforward bacteria and make that bacteria do the same thing? Is that a possibility?
Maybe. It would depend how compatible that gene is with those systems. What I think is probably a more likely scenario is that they'll mass produce these enzymes, and then maybe, say you truck in. I'm just sort of imagining this. You scale up the process. You make a heap of enzyme. You truck in the plastic. You dump the enzyme into the big vat, and it degrades it.
Cool. Okay, so you wouldn't even need the biological.
You might not. Yeah. Or because of the two-step process, maybe that wouldn't be so easy. But what these researchers said was if they basically introduce a heat treatment and weaken the plastic in the first place, and then add the bacteria, that makes them decompose the plastic a bit more easily. So that could be a possibility. Or you could just produce this all industrially and just say, well, here's my plastic processing plant. The first step is the heat treatment. The second step is this first enzyme. The third step is this other enzyme, and then you end up with a fairly harmless carbon source that you can probably use in anything you like feed, I guess.
So my first thought, of course, was the great garbage patches in the Pacific Ocean. These 5 or 6 clumps of plastics of huge sizes. Well, tiny sized plastics, but large clumps of them. So it sounds like this isn't the sort of thing that you can just sprinkle a whole heap of this bacteria on that and have it go to town. You'd really want to sort of collect the plastics, heat treat it, as you say, to break down that fibrous crystallized structure, and then approach it that way once it's
the other
broken
thing
down.
is that, remember, the bacteria will only eat something if they absolutely have to. So basically this procedure might be its last resort to get food. If there is another more easily accessible food source, it'll probably use that first.
Fair enough. So you just have to control for that.
Well, yeah, but what I'm saying is, so basically, if you just chucked, say, your hypothetical culture of Ideonella sakiensis on the big garbage patch in the ocean, it'd probably end up eating all the organic matter first. Which is
we don't want that.
Yeah. Which, no, to be honest, if there's a whole lot of stuff on the plastic, that might be a good thing. On the other hand, that would delay the process. So I suppose I think the industrial way is a lot more feasible in some ways. Just isolating the enzymes, sorry. But, yeah. It's really cool. I'd like to see where they go with this. I'd like to see if this actually goes somewhere. Because this is,
Well, I mean, this is just lab experiments so far, and there's nothing been done to scale it up.
No, but
that's the difficulty with this sort of thing is, okay, it works in a lab in a small scale, but when you try and do something on an industrial scale, it can be very different.
You'll come across all sorts of issues. The difference here is that this bacterium seems to be fairly easy to grow, which is the first stepping. It's your first problem when it comes to this. So if you can overcome that, then the next step, it's not simple, but it's simpler.
Cool. All right, Penny. A newly discovered horse-sized dinosaur reveals how Tyrannosaurus Rex and its close relatives evolved into the top predators of their time. It's all about brain size, really, isn't it?
Yeah, so when you look at the Tyrannosaur evolution, I mean, I would say almost everyone, if you think of a dinosaur, will probably think of Tyrannosaurus Rex pretty soon. Big elephant-sized, smart predator. But the family... And I don't know if this is exactly like a biological family, so please forgive me for not getting my terms perhaps exactly right. But the family, the Tyrannosaurus is a late Cretaceous kind of dinosaur, but the family itself evolved in the Jurassic, so about 170 million years ago.
Oh, so that's why it was in Jurassic Park. Sorry.
Anyway, the first Tyrannosaurus ancestors were quite small. And what I didn't know is, even though we know a lot about Tyrannosaurus ancestors and relatives and so on, there's about a 20 million fossil gap in the records. So we know what they were like from 170 million years ago to 100 million years ago. Nothing. And then at about 80 million years ago, they pop up again, and they're big, they're smart, they're top predators, Tyrannosaurus and all of its relatives. So this new fossils that's been described is not actually an ancestor of Tyrannosaurus Rex. It's like a cousin. It has a common ancestor.
Okay.
I don't know, 150 million years ago or something.
Yeah.
But it is in the Tyrannosaur family. It's in that gap. And what it shows is that they got smart before they got big. So it's horse-sized, not elephant-sized, but it's already got a really big brain. And one of the things that we find is that predators, carnivores, tend to have to be a bit more intelligent than herbivores. So
you've got to outsmart the prey.
You've got to outsmart the prey. Like, you have to catch it. You often need more intricate vision, which has a bit of mental machinery behind it, more 3D vision and so on. So I think that's interesting. I think it's what people would have guessed happened anyway, but there's such a difference between guessing how something would evolve and actually finding fossil evidence. And it's really important to actually find that evidence.
Yeah. And as you say, it's not that surprising. I mean, that's what we believe happened with Homo sapiens and the evolution of man, was that our brains got bigger, and to fill that we needed to start eating meat and getting higher energy source like that. And as our brains expanded and we got smarter, and we then were able to dominate the landscape rather than being just a bunch of chimps running around, which is cool. Cool. I don't know if there's anything else to say about that, except it's great that we've got that fossil record, because I do believe those gaps in the fossil record, I've got on very good authority, that proves that evolution is false.
Yeah. Oh, yeah, absolutely.
Yeah. So the more we fill those gaps, the more we solidify evolution as a theory or something. I don't know. Okay, let's move on and talk about what your parents ate before you were born, which can affect their children's health later in life. Shane, tell us all about the fat mice.
First of all, I just want to point out that Penny didn't want to do this story because she had mother's guilt, and I thought that was hilarious. Like,
Mother's guilt.
Oh no,
Too
I've
much
eaten,
mother
therefore my kids will be fat. Seriously.
Well, no, I think there is a legitimate element of that.
There is a legitimate
element. You look at
well,
all the advertising and all the way newspapers will approach a story like this. It's all about how what you as a mum should be doing to make your child smart.
And
all of that.
They recently changed the guidelines, and now I was supposed to have eaten 3 servings of oily fish a week all throughout my pregnancies and breastfeeding.
You have ruined your children's future.
Well, I was actually
You ruined
avoiding
their
fish
lives.
because of mercury. So,
yeah, I was going to say mercury, and obviously you're not allowed to eat like sushi because that's raw and that could like
yeah, exactly.
bad for kids.
Like, ugh.
Seriously,
You just can't win.
We've been having babies for hundreds of thousands of years, and most of them have turned out okay.
Yeah.
But a lot
I
of
think
them
she'll be
used
fine.
to die, Ed.
I've got nothing to say. I'm out.
Anyway, so I what's the word? I excuse myself from this story.
yeah, you did. I found it interesting because of the buzzword epigenetics.
Look, I like epigenetics too. So.
Yeah, so I won't even begin to try to understand fully the implications of epigenetics or even all the mechanisms of it, because they are numerous and complex, and a lot of times a lot of scientists are like, yeah, we found something else that could be epigenetic, and geez, there's so much of this stuff. Oh god, this is a complex system. Oh my god, let's go have a beer.
All right, well, let's first get a summary of epigenetics then.
That was basically it. No, it
wasn't. In no way did that summarize epigenetics. That just said that it's a buzzword that is used a lot.
So basically the dogma behind genetic inheritance is that you pass on your blueprint to your children, and whatever you do in your life won't necessarily affect them. So, for example, if you lose an arm during your life, your children won't inherit your lost arm, for example. Like, they won't be born armless. Okay? That's
so the genome is there.
The genome is there, set in stone. You get a bit of rearrangement at conception, and that's why we don't look exactly like our parents. But the general feel is that, what's in the DNA, it might mutate in your gametes or whatever, but it's not going. Anything you do in life is not going to affect that system. However, epigenetics puts a little bit of a spanner in that works, and what it does, it's basically editing the DNA on a cellular level while you're alive and changing it a little bit. Not necessarily the code, but things like molecules that are on the DNA that could act as signals for other mechanisms that can then be passed on to your children. So, for example, one very common form of epigenetics is methylation, and it's basically where they add something like a methyl group, which is like a little chemical group, will be attached to a strand of DNA, and that will act as a signal for something. It could be to say, ignore this gene, don't turn it on, or it could be to say, don't turn it on in vast quantities.
Mm-hmm.
So to simplify this even further, if a methyl flag is placed on the gene that says that you're full, and this flag is saying don't express this gene, you will keep eating.
Yep.
And you won't be full.
That's my excuse, and I'm sticking with it.
So that's a really simplified, possibly slightly incorrect way of looking at it, but that's one sort of way it could work. And then theoretically, you could pass that change onto your children, and they could then inherit this same problem. Okay? So that's sort of epigenetics in a really simple nutshell. Now, the way they looked at this was, again, a mouse model. So take this with a grain of salt. So they took 3 groups of mice, and they fed them 3 diets. They said, okay, here's the normal food, the normal thing that we feed lab mice, a low-fat diet and a high-fat diet. Of course, after a few, however long they fed these mice for, they found that, the high-fat mice are fat, funnily enough, more prone to things like type 2 diabetes, stuff like that. They then took the eggs and sperm from these mice and implanted them into normal surrogate mothers. Okay, so they've cut out the whole birth process in essence, and essentially given these mice pups, a new lease of life. What they found was that the pups that came from the group that had been fed high fat were more likely to also be obese, and the ones that came from the low fat diet, even if you fed them a high fat diet, were less likely to be obese. So this is where the epigenetics things come in. It's basically something has happened in between that, during the mouse's lifespan, it has passed on these changes to its children without, so its genome hasn't, that's what I mean. Like this, the Lamarckian evolution, which is the, whatever you do in life will then affect your children kind of thing, or the epigenetic way of life, epigenetic view, depending on what you want to call it. It's very interesting. And obviously the implications are here that if you are fat if you eat crappily as an adult, your children will also be fat. That's the very crude implication.
But is this talking more what they ate when they were what the mothers ate when they were pregnant, or is it
no
right up until pregnancy sort of thing?
It was because they took eggs and sperm from these mice, not the embryos.
So the mice weren't even pregnant.
They weren't even pregnant. They took the gametes and they used IVF to fertilize new mothers.
From a different group that's eating a different diet.
Yeah, I'm guessing they were the control group. Just the normal mice. So, they hadn't. That's what I find interesting about this. And I would love to read the actual paper, but of course it's
paywall. Probably not.
I'd like to.
Nature wouldn't do that.
No, of course I wouldn't. Look, it's a really interesting study. And
well, the thing about epigenetics is it is such a new field that it's just it's something that makes huge changes to our way of looking at genetics and biology when you have a new branch of it like this, I think. Yeah.
It's not even that it's new necessarily. I mean, it is newish, but it's also that it's so damn complicated.
Yeah.
And we don't really understand just look up the epigenetics entry on Wikipedia and it's huge. And it's very interesting. And there's all sorts of, this could happen, but we don't really know. You know what I mean? There's a lot of that. And it's definitely a growth area in science, and it'll be very interesting in the future. It's just very, very complicated and kind of daunting. But so the implication here is that, if you extrapolate this to humans, which you can't obviously do absolutely.
Because humans are not mice.
Yeah, A, and B, this is a very controlled study, whereas, there'll be a whole lot of other factors in play with humans. But the idea is that, if your behaviour as an adult can affect your offspring. It will change your DNA, and you will pass those traits on to your children, even though they originally were encoded in your DNA.
All right, very cool. Now, Lucas, you wanted to mention a recent discovery about a quasar because of the staggering numbers involved. This is about a extremely fast-spinning supermassive black hole, is it?
Yeah. This one's very cool, actually. It came in a week that sort of left me a little bit empty for stories, and I stumbled across this one, and reading through it, it was really the numbers that just blew my mind, and I love these sort of stories, and it also the aspect of it of how they they did these calculations and what they meant, which is just really cool. I love the sleuthing element in astronomy. So basically what's happened is they had this particular quasar, which is a type of supermassive black hole with an extremely hot accretion disk, which is all the material that's basically, falling into the black hole. It's feeding the black hole. So when you see one of these objects in the sky, typically they're observed, for example, in the X-ray band. They emit a whole lot of X-ray, which is a result of redshift and so forth as the light makes its way to us. They have quite a distinct signature, and it's the heat basically that's generated by what they're eating. This feeding event that they're doing, and this particular one that they had a look at, which is called OJ287, itself is around 18 billion solar masses, which is big. 18 billion solar masses. I'll just say it again. Billion solar masses. It's big. It's freaking big. It's really, 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 really big. So this one is
that's bigger than I thought. That must be one of the largest quasars we've detected.
I don't think it's particularly special in that regard. It's just supermassive black holes are well named. They are supermassive. Although this story suggests perhaps this might be one of the biggest ones that we've been able to get any kind of estimate about. Maybe it should even have its own class of an ultramassive black hole. But yeah, it's really big. But the thing is, we don't really know for sure how big they are. But big. So there was something that was interesting about this particular black hole, which is that, or this particular quasar, is that it had been observed to have these significant brightening events every 12 years. So that in itself was interesting because of what is happening every 12 years. So whenever you see something that's a repeated cycle, in the cosmos, you tend to start looking for orbital interactions because that's the most obvious, scenario here. But what would cause it to brighten so much? And what they've done is they have ascertained that the brightening event is actually two brightening events that are very, very close together. And from this information and a whole lot of other crazy math and measurements, they have figured out that there is actually another black hole, which itself is a mere 100 million solar masses. Just a piddling, next to its supermassive black hole. But 100 million solar mass black hole is going around this thing at quite an eccentric orbit. So it comes quite close to this ridiculously large black hole. And as it does, it dives through its accretion disk. So when it dives through, of course, it's a black hole as well. It suddenly disturbs this accretion disk and heats it up even more around it as it goes through. And then, of course, it loops around. If you can imagine a comet going around our sun, same sort of thing, loops around the back of the supermassive black hole and then comes through the accretion disk again as it leaves out and then goes basically out for 6 years and then comes back again. Really, really cool. Wow. So from this information, they further figured out that the spin rate of this supermassive black hole is something on the order of a third of the speed of light. So this is something else that is also special about quasars. We know that they will be spinning very, very quickly, but that's ridiculously fast. When you consider how large this thing is, it is spinning at a rate of about a third of the speed of light. So that's relativistic speeds. If I've ever heard them, that's insane. So if you can imagine something that big,
cosmic terms, can it keep that speed up for any length of time? Or?
Well, if it's feeding, yes. Typically, things get to ridiculously big speeds when they shrink. So, if you think of, say, our sun, eventually it will become just a white dwarf, but it will still have a whole lot of rotational energy. So it's that old scenario of, you think of an ice skater with their arms out, and then when they bring them in, they spin much, much faster because they've still got that angular momentum. Same sort of thing typically happens with these, with objects like this. But this already has that much angular momentum. It's ridiculous. So, eventually, but we do believe that, and this is something that Stephen Hawking contributed. We do believe that black holes do radiate off. They effectively evaporate over time, over a really, really long time, but they do evaporate. So eventually, this will get smaller and smaller and smaller. It's staggering to think what sort of speeds it would be at, after shrinking. That would just be that'll be amazing. And this is way beyond my layman's sort of understanding, but wow, the numbers really just blew me away. And they also said that at some point hopefully in the not too distant future, given the recent LIGO discoveries that we've covered on the show, that there would also be fairly significant gravitational waves being generated by these events. So given that we have now a model to predict it within the most recent prediction, because the last brightening happened on the 18th of November last year. So the dude who did the calculations had calculated it to within 2 days of when it should happen again. So we obviously understand this model very, very well. So that being the case, we now know where to effectively, point detectors and so forth for the next gravitational wave event when we've got better technology. Because at the moment, the LIGO detectors are not strong enough to be able to, or not strong enough isn't the right word. They're not. It
doesn't work on that frequency.
Yeah, but yeah, I mean, it's just the numbers are the thing that just blew me away. It's just awesome, and the fact that they're able to extrapolate all this information from what effectively is just something bright in the sky that brightens a little bit more sometimes.
Yeah, and I also thought just straight to the gravitational waves thing, because I remember when we were talking about that, we were even blown away that there were two black holes so close to each other and orbiting together like that.
Yes, because that was, we believe that to have been a merging of black holes, the detection.
Well, yeah, but it merges because it gets so close, and then as the spinning increases, they get drawn to each other, and then they merge.
That's right, yeah.
But, I mean, these are way bigger than the black holes that we're talking about with the gravitational waves that merge. This is incredibly huge.
How big is it? Yeah. Really big.
Wow, that's astronomy, and that's physics for you. You just get these incredible numbers sometimes.
I also like that it was almost exactly 100 years since Einstein had theorized a big part of what they used to find and understand these things, which is also nice. It sort of wraps around on itself. But cool story. There was another one that I almost talked about, which was also really, really big stuff. But this
you tease! You have to tell us that now.
All right, so there's a group of stars that have been found in the past. They're in the Tarantula Nebula, which is a part of the Large Magellanic Cloud. So if you look up at the Milky Way in a night's dark sky, you'll see sort of two blurry smudges off to the side of it, and they're the Magellanic Clouds, and there's a large one and a small one. So in the large one is a very beautiful nebula called the Tarantula Nebula. And inside that is a star that we believe, it is a star that is currently the largest known star. And this particular star was found back in I think it was about 1960 or something, that it was found through, I think it was through infrared detections. And that star, by the way, is about 100 solar masses. Hang on, is that right? No, 250 solar masses. So the really, really big star in this Tarantula Nebula, which is R136A1, is more than 250 solar masses, the largest currently known star. So that's big.
So when you say the largest known star, you mean the most massive?
Correct. Yes. Yeah. Good point. Cool. It is the most massive known star right now. It's of the class the Wolf-Rayet type of star, which have got their own funky stuff. But in this nebula, there's these are all. So the thing about large stars is they have very short lives. They grow fast. They burn incredibly hot. They push out a huge amount of material really quickly. Some of these stars are pushing out in the order of 1 Earth mass per month. That's how much they're pushing out in away from themselves. So they do burn their energy really, really quickly. But this recent change is a team pointed the Hubble Space Telescope and used its ultraviolet, its UV detectors to have another look at it, and they found there's a whole lot of other really, really big stars in this cluster that they didn't know about. And the reason is it's for whatever reason there's a big bunch of stars forming all at once. So it's a star forming region, which in itself is becoming a rare thing in the universe. So all of them together, and they share an area that's really close to each other, which is also kind of cool. But so it'll be another thing to keep an eye on is exactly what's going on there, because it's a really special area of the sky, And, relatively speaking, it's fairly close to us. It's about probably 170,000 light years away from us.
Right, which is pretty close, as you say. I mean, it's not like a trip down to the grocery store, but it's a long way away.
Yeah, yes.
No, I was trying to throw in the Douglas Adams quote there, and I stuffed it up. Oh, well. Very cool stories, though. As I said, huge numbers involved. Mind-boggling.
Well, yeah, I mean, those really big stars are in an area that's only a few light years across. That's pretty mind-blowing in itself. You wouldn't want to visit, really.
I'll cross that off my bucket list.
Yeah.
All right, that's our show. Links to all of those stories are on scienceontop.com/219. There you can find our social media links and ways to get in touch and leave some feedback. You can always email us feedback@scienceontop.com, and don't forget to leave a review or rating for us on iTunes. We're very grateful to Addicted to Piano for writing a very nice comment on our YouTube channel for episode 217. It's nice to know we have a fan in Germany, and a physics student, no less.
Nice to know we have a fan.
That's great too.
I'm just glad that we haven't, obviously, if they're a physics student, we haven't stuffed things up too much. So.
Oh, yeah. After that whole schmozzle with the how big is this star and everything, he's probably just lost it with us now. But it was nice while we had you addicted to piano. Interesting name. And as always, thank you, Penny, Shane and Lucas.
Always a pleasure.
No,
thanks, Ed.
This episode was edited with cleverness and zeal by Marcos Benamu. And thank you, everyone, for listening. We're going to have a short break over Easter, but we'll be back the week after, putting science on top of the agenda. Join us then.